is it that almost every cell in your body has the exact same genetic information, the same DNA blueprint, but they do totally different things? How come your toes are different than your nose? How can you grow hair out of your head, but you're not growing hair out of like your fingertip? Basically, the short answer is it's complicated. The long answer is it involves a lot of different levels of regulation. So basically, although all those cells have all that DNA, there's so much DNA that they have to actually like coil it up really tightly. And so it's actually hard for your cells to even get at those instructions to do various things because that DNA is tightly coiled around these proteins called histones. We'll talk more about this. But basically, if a cell wants to express a gene, so basically use that recipe, they're gonna have to unwind that DNA. They're gonna have to push those histones out of the way, unwind the DNA, and actually use it through transcription, where they're gonna make copies of it, and then those can be used to make protein from it. So there's a lot of transcriptional regulation as well as epigenetic regulation. So basically, epigenetics is above the level of the DNA. So in the DNA is written the instructions for doing things like making proteins. And that those instructions are then going to be tightly coiled up around these histone proteins. And in order to better like tell cellular machinery, okay, well, come to this place and use this gene right now. Basically, you have to remember, move those histones out of the way, kind of. And so you can modify those histones with like acetylation and methylation. So this is various epigenetics. It's epi above the level of just the DNA, just the gene sequence. And so this is an extra level of regulation that's going to help regulate like when the regions are even accessible. Now, when the regions are accessible, that still doesn't mean that they're going to be used. And so in order to actually be used, you have to recruit the machinery that's going to do that transcription, that's going to basically make a messenger RNA copy of that DNA that can then be used to make protein from it. And not all proteins code for proteins, but we're just gonna go with that for now because it's simplest to talk about. But some make like functional RNAs that work as is and don't need to be like translated into proteins. But basically, most of the time when we're talking about like protein coding genes, so instructions for making proteins, you make a messenger RNA copy of that, and then those copies are then exported out of the nucleus. So I didn't mention, but all of this is happening in this membrane-bound compartment in your cells called the nucleus. It's kind of like a restricted section in the library, so you can't check books out of there, but you can make copies of them and take them out. And so this is what your cell does when it wants to make a protein. It makes copies of the DNA recipe, ships them out of the nucleus into the general interior of the cell called the cytoplasm, where the protein making can occur. But in order to make those messenger RNA copies, you need to transcribe the gene. And so we have to open up the region, and then we have to get the transcription machinery, this RNA polymerase, to actually come and bind in that front of the instructions and actually make the copy of it. So in front of those instructions, there's a region called a promoter. And there's also like regions like enhancers and things like this. But this region is going to be where the RNA polymerase is going to get recruited to, and it's going to start making its copies. Now, in order to recruit the RNA polymerase, you can kind of have transcription factors that can basically kind of typically proteins that are going to bind, and they can help like bind to the polymerase or activate the polymerase or various things like this, recruit all the other machinery that's needed in order to get this process going. And so we can have these transcription factors that can then help get this going. And it's not that there's a single transcription factor that works for that every protein has, or that a single protein has just one transcription factor. Instead, it's kind of mix and match. So a protein can be regulated by a variety of transcription factors. So they can have binding sites upstream of the gene for a variety of different transcription factors, and this allows them to be regulated by different transcription factors. Additionally, they can have like multiple proteins can have a binding site for the same transcription factor. And this allows you to get like combinatorial regulation, and it allows you to basically activate this a set of multiple genes with the same transcription factor. So this allows your cells to kind of have presets. So it's like kind of in your computer program, you might have a preset where you run different programs at the same time, or you do these things at this time. If I press this button, and if I press this button, do these things. Basically, in your cells, you can have like, okay, well, if this transcription factor is made, now make these, or now make these. And so now your different cells just have to control those transcription factors, and they can express different transcription factors in different cells. And often when these transcription factors are regulated by is like external signals, so things like hormones and other chemical messengers. You often get these messengers binding on the surface of the cell to receptors, and there are different receptors for different types of chemical messengers. And you can also get messages related like directly from a cell to a cell through like contact and things like this. And those messengers can relay the signal into the cell. So often you have the messenger like bind to the, so we call it like a binding partner, a ligand. So you have a receptor's ligand, its binding partner. So this can bind to the outside of the cell on that receptor, and then the receptor kind of shapeshifts. It undergoes what we call a conformational change or a shapeshift. And this kind of changes the inside part of that receptor. So the receptor is going through the membrane, and then on the inside is the inside of the cell, it undergoes some sort of like, it changes shape, and this can like activate a kinase or something like this, which is basically a protein that adds a modification called a phosphate group onto other proteins. And this can cause those proteins to then get like activated and stuff. And so you can set off these signaling cascades inside the cell that lead to the transcription factors being like made or being recruited to the various sites. And then once those transcription factors are present, then they can go and they can actually, so sometimes they're also like hanging out in the cytoplasm and they get imported in when it's time to go, when they get the signal. And then some like hormones, like steroid hormones, can actually like go into the nucleus, various things like this. Complicating factors I'm not going to get into. Bottom line is these signals can lead to transcription factors being like recruited to various genes. And you can have the same transcription factor, remember, be activating multiple genes that are related that you need to express at the same time. And so your toes can express different patterns of genes than the cells in your nose. And both of these are then able to change what they're doing based on the cellular signals that they're getting from outside. Now, sometimes your cells your cells, they all start what you might have heard of like stem cells. So these are cells that have the potential to like differentiate into different types of cells. So they start off as like these cells that are like have the ability to become all sorts of different types of cells. But then they start making choices and based on like chemical signals and things like this. And early on, they're going to kind of silence various genes. So this is going back to the idea of like epigenetics and stuff. So sometimes they make it so that they kind of like wind up parts of the DNA and like hide it that they're not going to use. And then the other regions is more dynamic about like whether or not you're using them or not using them. You can kind of open or not open these cellular regions of the DNA to use, and then some regions that are used a lot are going to be more open. We call these like euchromatin versus heterochromatin, where it's really, really tightly wound up. And so your cells can regulate the accessibility of various regions and then their usage. And so in this way, by like early on, your cells can then like set out on different fate paths, which is going to influence what transcription factors they're like making most of the time that they don't need like a signal to respond to. So there's various ways that your cells are smart and are able to be different types of cells and have different options. And there are also, in addition to like positive transcription factors, there are negative transcription factors, or that are actually going to like repress the usage of a gene. So you can repress the usage of a gene through ways that are like epigenetic methods that are going to kind of tighten up the chromatin. And then you can also have regions that are going to, you can also have regulation based on like having transcription factors that are going to repress the translation, or transcription, maybe they hide the binding site for the RNA polymerase or things like this. So you can have, and it said then you can have a combination of positive and negative regulators. So there's a lot of different regulation that can go on, so your cells can integrate multiple different signals. So, as I said, it's complicated. And one more complicating factor, I just wanted to mention, because in the beginning, you know, I said like almost all of your cells have all the genetic information. Well, there's some cells in your body, like your blood cells, the red blood cells, that they actually kind of like ditch their DNA because they don't need it. They just like use it to chalk up full of a bunch of like hemoglobin off, mostly. So this like protein that binds to oxygen. So those red blood cells have a ton of hemoglobin, but they don't need the DNA anymore, so they kind of like ditch that. So your mature erythrocytes or red blood cells actually don't have that DNA. Now some cells in your body actually have multiple copies of the DNA. So like your muscle cells are like multinucleated. So the nuclei, remember, that's where all this DNA is, and so your muscle cells have more of it. And then you also have like immune cells that have a slightly different DNA because they actually edit their DNA. It's like somatic hypermutation and things like somatic mutation, somatic hypermutation, all these various like immune things, where basically they have mechanisms that they can change up the regions of DNA, kind of like swap out different regions. And this is how you can get different like antibodies made by your immune cells. They kind of do this like cut and paste and kind of in like the B cells and stuff, which I'm not going to get into, but just know that that's a process that occurs in these immune cells that's not happening in other cells in your body. And then the DNA in each of your cells is slightly different just because of like random mutations that are always happening. So there's just going to be like heterogeneity. So basically just like differences between these different, the DNA in these different cells. Most of the time these differences aren't even consequential. And most of the time they get fixed, and even if they don't get fixed, it's really only in your germ cells, so the ones that are actually going to be used to make like sperm and eggs and stuff that would get passed on to a future generation. So if you have like a mutation in a somatic cell, so one of your bodily cells that's not like one of those germ cells, it can only get passed down to future cells in that lineage, so cells that are made from that cell. So this can still have important consequences, such as like causing cancer and things like this, but you still have a lot of like generic, not generic, but just low-level mutations that are not consequential, because not all mutations are harmful. And some of this is because it's not happening in those regions of the DNA that actually has instructions for making a protein. Sometimes it is, but it's still like not consequential because it doesn't actually change the protein letter. Much more on this in other posts. But basically know that mutations happen a lot. Most of the time these are going to get fixed. Sometimes they don't get fixed, but usually it's still not a problem. And so you're going to have variation between your different cells in your body, but this is just like a low level of variation, and it's nothing like the variation between people. And yeah, so that is the basics. And so bottom line is different cells in your body are going to use or like express different genes at different times. So although all of the cells in your body, or almost all, as we talked about, have the same genetic information, the same genetic blueprint, they're not always using everything. In fact, they're only using like a tiny portion of it. And different cells in your body are going to be using different tiny portions, and they can change up what portions they use at different times. It's kind of like how a restaurant might make different recipes in the summer versus in the winter. But the summer in like California is going to be different than the summer in Australia. So summer is like now in Australia, but here it's like all rainy and cold. And so different places in your body are going to make different things at different times, and they're also going to have different recipes that they want to use. Like, I'm not eating Vegemite. So basically different cells in your body can do different things, and the way that they're able to control which of these different things that they're doing is through various levels of regulation. And so the main level of like turning genes on and off is basically by transcribing them or not transcribing them. So transcription is the process where you make a messenger RNA copy of a gene that can then be used to make a protein. And we can also remember have like functional RNAs that are going to act at the RNA stage. But both of those are going to require making a RNA copy of the DNA, they're going to require transcription. And whether or not DNA can be transcribed depends on whether that DNA is accessible. So is it tightly coiled up around those histone proteins in heterochromatin, or is it more accessible? Is it in euchromatin, so these like freely accessible regions? That's going to be dictated by epigenetics. So basically you can have those histone proteins have various markers that are going to make it so that they're more open or less open, that they're bound more tightly or less tightly. And so this can be through like acetylation and methylation and things like this. It's not quite this simple, but acetylation, basically it acts as the charge on the lysine, makes it so that it's going to be looser. So acetylation is often, it loosens up the histones. They have these like tails, which have a bunch of these lysines, which is positively charged, and DNA has negatively charged backbone. So this is going to make it so that those histone proteins are going to be more tightly bound to the DNA. The DNA is going to be tied up more tightly. It's going to be less accessible. If you acetylate that lysine, which is a modification you can add to the lysine, it's going to then make it more open because the lysine is going to not be charged anymore, and so it's not going to be as tightly attracted to the DNA. And methylation marks, these don't change the charge, and they actually make it so that the DNA kind of like is more tightly bound, and it also offers binding sites for other proteins. And so methylation and acetylation, in addition to just changing how tightly the histone tails are bound, like the histone proteins are bound, it can also add binding sites for various transcription factors and other regulatory factors. So these are epigenetic marks, and you can also have epigenetic marks on the DNA itself, like DNA itself can get methylated, in addition to the histones being methylated. So those are a variety of epigenetic modifications that can regulate like how accessible the DNA is and when it's used. And when it's used, if it's accessible, it still might not be used. In order to be used, you've got to get that transcription machinery recruited there and get it going. And so this is where like promoter regions upstream of the gene come into play, and how you can have transcription factors that are like typically proteins that are going to bind in that promoter region and like help recruit things or keep the thing like RNA polymerase from binding or things like this. And you can have different combinations of transcription factors in front of the same gene, as well as the same binding sites for the same transcription factor in front of multiple genes that you want to regulate at the same time. And then those transcription factors, their like availability and their making of them, and then how available they are at those sites, is also going to depend on the things that are happening externally outside of the cell, like chemical signals, like hormones and things that can then send signals throughout the cell and activate these various pathways. And because it's relying on these signals, by changing the signals, you can then respond to different circumstances. And then the other thing is that cells can early on kind of make it so that they make regions of the gene more or less accessible. They start out setting off like cycles, like positive feedback and negative feedback and things like this, to help regulate which of the things that they make like most of the time, and how much of those things they make most of the time. And then they can kind of just regulate the levels of the other things. And so there are other ways too. Once you've transcribed that, you can regulate at the translational level. So how much of the protein is how much that messenger RNA is being used to make proteins, as well as how much of that messenger RNA actually sticks around, so we can make multiple copies of it, and then we can degrade those copies, and we can also modify the proteins that are made. So there's a lot of different regulation that can occur, but the first step for all of that is transcribing the gene. And this is where you can get those really, really big differences between different cells, is whether or not these genes are kind of like used at all. So hope that helped explain things. And yeah, so yeah, a couple of people had asked about that, so I figured it was easier to just make a quick video. So hope it helped.